We are very lucky today to have Dr. Tim Alcorn with us. Dr. Alcorn has experience as a clinical chemist and a molecular biologist. He has worked with research labs across the globe. He's a world traveler, which I think is really cool. He's worked with medical device design. He's worked with tests, field tests. He's worked with, and I didn't realize this, in vitro medical device manufacturing. But he's been in healthcare for, gosh, almost 30 years. He's one of the smartest people I know. He just so happens to be my brother-in-law. And I'll share two personal things about him. One is that he's an excellent cook, and he has cooked fish in a dishwasher. And another thing is that when he travels the world and he's in a different country, he has been known to ask the waiter or waitress just to bring him the most interesting, authentic thing on the menu, and whatever it is, he eats it. So I think that gives you a little bit of a feeling for who he is as a person. And I think you're going to be very impressed with who he is as a scientist. And with that, Dr. Alcorn, please take it away.
Sure. Well, thanks, Steve. And yeah, so you forgot to mention that I am from Lynchburg originally and graduated from Heritage High School. So kind of the same place I'm talking to. So I appreciate the opportunity, and I appreciated the questions that you guys submitted. They were all right on point, and hopefully we'll get to as many of those as possible. Let me just give you kind of a brief background on myself and what I normally do, and then we can dive into your questions. So as Steve mentioned, I am a chemist by training, a clinical chemist, but I spent most of my time in infectious diseases. I was the head of molecular infectious diseases for LabCorp for over a decade, and then the head of clinical affairs for Janssen Diagnostics, which was part of Johnson & Johnson for many years. But most of my time has been spent on infectious diseases and mostly in virology. So my background is heavily focused on diseases like HIV, hepatitis C, other virological diseases. And I am currently at the Latham Biopharm Group, which is a consulting group, and we help companies and bring new technologies to the market broadly across the different types of medical products that come onto the market, with a heavy focus on public health. So I spent many time working on vaccine development, on therapeutic development, on analytical testing methods, both for the developed countries like the United States, as well as underserved countries. We do a lot of work with the Bill and Melinda Gates Foundation and some of their efforts to look at global public health. And of course, when COVID-19 happened, most of the people I work with are heavily in infectious disease. And so all of our efforts have been focused on finding solutions for this pandemic, both therapeutic solutions and vaccine solutions, as well as diagnostic solutions. So hopefully I can answer some of your questions today that you posed, and we'll jump from here to whatever the first question is. So please fire away.
I was wondering, what were you working on previous to the COVID-19 outbreak?
Previous to that, it was a variety of things, including malaria, HIV, bacterial infections, also looking at things like autism, cardiovascular disease, so a wide variety of medical diseases and conditions.
Next question.
What does your daily schedule look like as you work through this pandemic?
So most of my time is spent writing up information that is used to propose ideas for federal funding. So I I outline clinical trials that are used to test new drugs and vaccines. I write up rationales for new diagnostics. We also do a lot of time spent analyzing data. So we get large data sets in from places like Wuhan, China, from the Bronx in New York. And so we look at that data and see if we can make clinical findings out of that data. So most of my time is spent reading literature and writing.
We were just wondering, like, what is your lab doing specifically to combat the COVID-19 pandemic?
Right. So I work with a bunch of different companies and laboratories that are developing products for COVID-19, and those range from diagnostic solutions to therapeutic solutions, vaccine delivery solutions, so a couple of vaccine projects, a couple of therapeutic projects. So it ranges across the board. And so we're working with companies in the US, but also outside the US. So we're working with a group in Japan and a group in the UK. So it's a wide range of different areas that are providing products to combat the pandemic.
My group was wondering, in your opinion, how is COVID-19 different from other pandemics you've worked with?
Right. So this virus poses really two primary challenges. So first of all, let me say that it is a coronavirus. So we know how coronaviruses work. We have looked at other coronaviruses, SARS being one, MERS being another. These are all strains that had high mortality rates. We've dealt also with, the common cold can be caused by a coronavirus, what we call the common cold. So generally speaking, what normally happens is when a virus is in the population, there's some level of protective immunity. So when the flu season happens, even if it's a new flu strain, typically there's some level of immunity that people have to that virus. So this particular viral strain is different in that there's no existing coronavirus that's similar enough that it provides any protective immunity. So that it's new to the human species, so there's not an existing protection system. The second is a lot of the pandemics, if you look at like Ebola, for example, things that have high mortality rates generally have less effective spread from humans to humans. It's sort of a self-limiting disease. This is unique in that its mortality rate is relatively high compared to, say, the influenza virus, but not at the level of, say, a SARS or a MERS or an Ebola. Therefore, there is a much greater risk that there is transmission of the virus, especially from patients who are asymptomatic or have mild symptoms. So it's highly transmissible. These two things combined is really what results in the pandemic we're seeing now. Next question.
All right, I guess I will ask the question. My name is Andrew, and our group was wondering if there was anything structurally different about coronavirus that compared to like these other viruses that makes it like more contagious, I guess.
Yeah, so it's a good. All these are great questions. That's a particularly good question. So the reason this virus is so easily transmitted is it has adapted itself to use a very common receptor on human cells, the angiotensin converting enzyme receptor, ACE receptor. So that is a particular. That particular mechanism allows this virus to be transmitted pretty easy. So all viruses have unique systems in which they can attach to human cells and how easily they're transmitted. So how well that virus can find those receptor sites to infect a person really dictates how infectious it is. This particular one uses a very common receptor on human cells that normally is used to control blood pressure. Next question.
So in regards to like basic vaccine manufacturing, I was wondering, how do you know which chemical and biological agents to use in a vaccine?
It's a great question. So one of the things I think that is incredibly encouraging about where we are today in science and how quickly we've responded to this pandemic is that we are now very good at figuring out where to look for vaccine targets. If this pandemic had happened, say, 20 years ago, we would be in much worse shape than we are today. So in real time, as soon as the virus was discovered in China and they were able to sequence the virus, in other words, they got the genetic sequence of the virus, people were able to design vaccine targets within a few hours. And the way we do that is we know structurally how viruses are formed based on their genetic sequence, and we can identify surface proteins that will exist on those viruses, especially classes of viruses where we've known before. So we've had other coronaviruses where we've developed vaccines against, like MERS and SARS. So we were quickly able to identify what the surface proteins of the virus look like. And vaccines have to be targeted to things that are on the outer surface of the bacteria, of the virus, so that the antibodies in the host, in this case, our antibodies, can find the virus. So what we do is we look for those surface exposed proteins, and then we start manufacturing replicas of those proteins and see if we can induce an immune response first in animals, and then hopefully that goes on to humans. So it's all done mostly through computer analysis and computer modeling of those surface proteins. Next question.
Hi, I'm Anna. So what are some common challenges that you face when your team is developing a vaccine?
So when people are developing vaccines, and this goes, I think, to another question, let me kind of walk you through the process of vaccine development. So the first thing that happens is you identify these potential targets that are on the surface of the virus. The next thing you do is you have to create a vector. We call it a vector. This is an artificial means to create that protein on the surface of something that we can use to see if we can elicit an immune response. A lot of times these are bacteria, or they're other viruses that we know how to work with, or sometimes they're artificial surfaces like latex beads. So you have to test multiple of these proteins, hundreds of these proteins, to find ones that conform to the right way on these vectors, such that you can elicit immune response. Then what happens is you have to test to see if that works by first looking at the immunogenicity of the vaccine in an animal model. Almost everybody uses rabbits as their first test subjects because it's very easy to isolate antibodies from rabbits. They have a very powerful immune system that produces antibodies quickly. And you can inject the rabbit with your vaccine and see if you can get antibodies against your target. Now, those antibodies are just the first step, because that means that now we have a possibility to elicit an immune response. We now have to test to see if it's effective against the target we were looking for. So for COVID-19, the first thing people do is they inject into rabbits and see if they get an immune response. And then they go to other animals. Rodents are typically used that we can first infect with the virus, and then we can test to see if the vaccine works in those models. The particular challenge with this virus is there really are no good animal models yet to do that second step, because it uses a very unique receptor we talked about before, the angiotensin converting enzyme receptor. Normal animals don't have this, so we have to make genetic modifications to animals to test it. But we can skip that phase if we've tested the vaccine in with other viruses. So a lot of the vaccines you see that are going into humans, they've already tested that vaccine based on a previous coronavirus like SARS or MERS. After the rodents, then what would be a normal process is you would go into non-human primates, which are the closest to humans, so monkeys, chimpanzees. If all that works, you've proven that you can, in animals, be safe and effective with your vaccine, then the next step is that you go into humans. And the phase 1 trials for humans, which are started already for some of these vaccines, is that you, in a very short time, a period of about a month, is you inject a normal, healthy, young human adults and make sure that they can elicit an immune response and that the vaccines are safe. That normally takes about a month. The second step, phase 2 of vaccine development, is now you go to a larger set of humans, generally in the hundreds, where now you want to see if the immune response is produced over a longer period of time. So these trials normally take about 6 months. So you inject the humans, you see if you need a booster shot, as you normally do with vaccines, and in about 6 months you can see if the immune response that you're getting from the vaccine is lasting. The third step then is the most difficult for vaccine development, and that is testing to see if it's effective at preventing people from getting the disease. These trials normally take thousands of volunteers to do, and what you do is you inoculate a community of volunteers, and you kind of know what the infection rate is for any disease, and you see if the patients who have the vaccine versus a control group that don't, what fraction of them get infected. That process for something like this could take 6 to 8 months. So when all put together, to prove that a vaccine is safe and effective normally takes about 12 to 18 months once you start human trials. That's the biggest challenge, is proving efficacy. Next question.
You basically answered mine in that one right there, so we can move on to the next one after that.
Okay, great.
Hey, how much longer do you think it'll take for you to come up with this vaccine?
Well, so there are multiple groups working on vaccines right now. I think 2 or 3 are in phase 1 trials. Not only in the US, there's trials happening in Australia and Israel and Japan. There's a good chance that within the next 12 months, we'll have a vaccine that's ready.
Okay, thank you.
Mm-hmm. Next question.
Okay. Our question, my name is Aaron, and our group's question was, there are joint computing projects that sequence proteins for different illnesses. I think generally not in pandemic conditions, it would be things like cancer and whatnot. An example of that would be folding at home. How is the data from these projects actually used? Like, is it for developing a vaccine or medication or like, yeah.
Right, so this is the general field of what we call computational chemistry. And so this is, it would be normal that most pharmaceutical companies, most vaccine companies have divisions of folks who are computational chemists. and they work with medicinal chemists, which are synthesized compounds. And generally, those folks are involved in the very early stage identifying targets for either drugs or vaccines. It's a robust field of chemistry at the moment, computational chemistry. And what they do is they take genetic sequence and protein sequences and have fairly elaborate software packages that can help them derive 3D models of proteins. And then they can look at 3D models of either receptor sites for vaccines or drugs, conformations of drugs, and see if they interact with the target of the drug or vaccine. So they're a very powerful tool in early phase discovery of new drugs and vaccines. Next question.
Are there any suspected long-term effects of COVID-19 or the treatments or vaccines? And how long until we're going to know if there are those side effects?
Yeah, so we won't know for the vaccines long-term effects, but we can for many years, but we have never really seen any long-term effects from vaccines. It's very rare that a vaccine has any adverse events associated with them. There are occasions that they do, and for rare, occasionally that people have trouble with vaccines, but that's rare. Drugs go through extensive safety testing. We know pretty much when they're released what their long-term ramifications may be, but those are closely monitored as they're released and start using. As for the COVID-19 itself, we don't really know, right? But there is indications that there may be long-term lung damage, and from recent studies, there may be cardiovascular damage that's occurring, and those may have chronic effects. But it's probably too early to know what that actually looks like.
And even if you isolate the virus, like if you create a successful vaccine, how will you be able to mass produce that? And would you be able to make enough for the population that's getting the disease?
Right. So that's a good kind of picture of what the world does in preparedness for pandemic events. So to produce a vaccine, what you do is they're normally produced through bioprocessing. So there are large vats that grow bacterial cells, that grow human cells, whatever is used to make that vaccine. There are a lot of companies that have this capability. In addition, the US government several years ago in preparation for the event of a pandemic flu event, developed several large bioprocessing manufacturing plants that are capable of producing tens of millions of doses a year, hundreds of millions of doses a year. So those sites are ready and ramping up, that once we have a vaccine identified, we have the scale to grow these vaccines and mass produce them. So we did a very good job of preparing for this type of event. Next question.
Hi, I'm Drew Flint. And my group was kind of wondering, how is the COVID-19 pandemic going to affect healthcare moving forward? So are we just going to like return to normal, or are there going to be some long-term ramifications?
There may be some changes to healthcare going forward. It's probably too early to tell. I think on the good side, and many of us have talked about this, this is kind of like the efforts in the 60s to go to the moon. We got a lot of new technologies out of that effort. Things like Mylar and things like that came out of the lunar efforts. The same thing's going to happen here. You're going to see a lot of very inventive new technologies that are ramping up at high speed that will come in to fight the COVID crisis that will have long-term positive effect on healthcare. So I think that's for sure going to happen. Next question.
Good morning. Our group was wondering, how as the clinical chemist is your work inhibited or otherwise affected by regulatory agencies such as the FDA?
It's not really. We work, pharmaceutical companies, diagnostic companies work very closely with the FDA. Of course, the FDA puts barriers in place that take time to overcome, but those barriers are in place because they want to make sure that things are safe and effective. So we don't really view them as challenges or barriers. They're more partners, and not only the FDA, but all of the regulatory bodies throughout the world, I think, have always recognized that their job is to be partners and not be obstructionists. So that's happening on a day-to-day basis. So we view them as partners instead of barriers. Next question.
Well, I think that is the end of the student questions. I have a question, Dr. Alcorn, if I may. Is there any effort being undertaken to develop a test to determine if someone has already been infected and has recovered and therefore would have an acquired immunity and could perhaps serve in the healthcare industry without worry?
Right. So there are 3 types of tests that are applied to this pandemic. The first type of test is test to see if you have the infection. And those tests are molecular tests. We look for the presence of the virus. And those are the tests you see really being used right now. These are the tests from Abbott and LabCorp and places like that. These are all PCR based, so they're all DNA and RNA based assays. The second class of tests is what you mentioned, and these are called serology tests. So these are tests that look at the host, the human immune response to infection. There's typically 2 types of immune cells that we look for. Immunoglobulin G, IgG, which occurs when someone is in acute infection, and then IgM, which is a convalescent response. So that is the one that occurs about 14 days after infection and is the one we look for to see if there's protection against future infections. So what you're going to see in the very near future is there's going to be this effort to screen large swaths of the population to see who was actually previously infected and never diagnosed. They will have positive IgM responses, but if they're recovered, they will have negative IgG responses. So we can use this to see at what stage of infection people are in. And I'll just mention the third type of test are not geared towards the virus itself or the host response to the virus in terms of the immune cells. The third type of test look for the disease severity. So the big question with this virus is we want to know when someone is sick, who are the ones that really need to care immediately? Who are the ones that we have to reserve ventilator space for? So what is this disease severity? So this is one where I've spent most of my effort recently on, and we're developing an app that will be published soon that will help doctors determine disease severity based on cardiovascular complications from the disease.
Thank you very much. Dr. Alcorn, would you have any closing comments for the students before we wrap up?
Sure. First of all, thanks for the opportunity. They were all great questions. So sorry we couldn't get to all of them. When you look back on this, and in fact, several of us were talking about this recently. one thing that you should take out of this whole pandemic is how rapid we were able to respond, and it's really an indication of how far science has come. We've had tremendous response from the scientific community coming up with very inventive and creative ideas to respond to this. I'll just tell this one, how big a response this has been. So there's an agency in the United States government which is designed to help with companies develop products for preparedness. So this agency is called BARDA, and BARDA normally in a normal year gets several hundred companies coming to them with new ideas about how to prepare for things like influenza or radiation or things like that. When the pandemic happened, BARDA had more than 4,000 companies come to them with new ideas. And the money that you see that the federal government is directing towards COVID-19, a lot of it's going through that. So this is really the moon launch event for this generation. You're going to see amazing new technologies that come out of this, and the long term is going to be a much healthier society when we get past this pandemic.
Thank you very much, Dr. Alcorn. I'm going to switch the recording to gallery view. So students, if you could thank Dr. Alcorn with a reaction for his time today. He's a very busy man, and I'm very grateful that he gave us some time to share this information with us. I will also mention that I am hopeful for next year when we're actually in person and you all are seniors, that I'll convince Dr. Alcorn to come to the Governor's School in person so you can thank him in person and he can share an update on the information that he shared today. So thank you very much, Dr. Alcorn, and thank you, students, and I hope everyone has a fantastic day.
Thank you. Have a good day.